Right now, we are going to have Raehoon Jeong. He's a PhD candidate working on bioinformatics and integrative genomics at the Bolaki Lab at Harvard Medical School. He also completed his bachelor degree at Swarthmore College in Pennsylvania, and he's going to talk today about using GWAS and different models to study non-coding regulatory genes and their variants in blood cells. So thank you so much, and go ahead.
All right, so I'll start sharing my screen.
Perfect. I
It
can
looks
see
good.
it.
Okay. All right. Oops. So to begin with, I'm happy to say that I successfully defended my PhD between the talk schedule and now. So I'm a PhD now. And thank you very much for the opportunity to present my work that I did during my PhD. And now we're going to switch gears to think about how variants in the non-coding regions can be interpreted for their function and their roles in various traits. And this work involves prioritizing causal non-coding regulatory variants associated with blood cell traits by incorporating transcription factor binding variation data. Hematopoietic stem cells, shown at the top, can differentiate into multiple types of blood cells. Blood cell measurements like lymphocyte count vary between individuals, and they are routinely measured as biomarkers because departure from a normal range can be a diagnostic indicator. And we have learned that variation in blood cell traits between individuals have significant genetic underpinnings. Through genome association studies, or GWAS, hundreds of blood cell trait-associated loci have been discovered. However, there are many remaining challenges in figuring out what the associated genetic variants are doing. First, GWAS loci generally have many correlated variants. In the toy diagram on the right, there are 5 variants that are strongly associated at similar levels. But this does not mean that they're all causal for this trait. The correlation structures between variants, namely linkage disequilibrium, lead to correlation in the association statistics. So pinpointing the causal variant is a major challenge. In addition, variants that are associated with many complex traits, including blood cell traits, are mostly non-coding, especially so for common variants with allele frequency greater than 1% or 5%. The functions of these non-coding variants are harder to predict than those of coding variants. For coding variants, we know how the amino acids are encoded in the mRNA sequence, but the non-coding region is vast, and we do not know just from the sequences what the functions might be. So these two properties make pinpointing causal variants at GWAS loci very difficult. Still, we want to know which variants are causal because we can learn about how the variants may impact gene regulation, and also we will be able to predict genetic risk in individuals more accurately. And through this project, I wanted to address these two questions. First, what is the causal variant? And what is the functional effect of the regulatory variant? Now let me introduce transcription factors, or TFs. They're sequence-specific DNA binding proteins. For example, here's a TF called PU1, and PU1 likes to bind sequences visually summarized by the sequence logo here. And this is called the binding motif. So for transcription factors, when there is a stretch of DNA sequence, PU1 can recognize its motif sequence and bind them. And when there is a motif-altering variant, that interaction can be disrupted depending on the degree to which the affinity is changed. Transcription factors are important because they regulate transcription. When TFs bind to enhancers and promoters, they recruit other proteins that set the stage for transcription. The sequence-specific nature of TFs and their role in transcription makes them a likely mediator of non-coding variant effects. That is why trait-associated non-coding variants are typically expected to alter gene expression via alteration of transcription factor binding. As a strategy to resolve causal non-coding variants to blood cell traits, I had the idea to focus on variants that alter the binding sites of a specific transcription factor. And a good candidate to study is a hematopoietic master regulator, PU1. As I depicted on the diagram, PU1 is up or down regulated in progenitors to direct the cells to various lineages. But it doesn't act alone. However, PU1 is one of the most important genes. So I investigated whether there are variants associated with blood cell traits that alter the binding site sequences of PU1. Then we need to identify the genetic variants that modulate PU1 binding. For this purpose, I analyzed PU1 ChIP-seq data from 49 genotyped individuals to identify PU1 binding quantitative trait locus, or QTLs. And just like GWAS, there can be many correlated variants with similarly strong associations, making it seemingly difficult to know what the causal variant is just by the association statistics. However, we know that PU1 prefers to bind its motif sequences. By analyzing the sequences surrounding the genetic variants, we can pinpoint a variant that alters the PU1 binding motif, and this is likely to be the causal variant. Now, the last piece of this puzzle is figuring out which PU1 BQTLs share genetic signals with blood cell traits. Colocalization methods specifically evaluate this. And under the hood, they compare the genetic associations in a locus for the two phenotypes, in this case, PU1 binding and some blood cell trait, and determine whether the data are consistent with the two phenotypes sharing the causal variant. Loci showing colocalizations are cases where a genetic variant likely affects both PU1 binding and some blood cell trait. In this example, the causal variant is associated with PU1 binding. So my hypothesis was that we can identify a variant that alters the PU1 motif sequence. Since this variant is causal for PU1 binding, it is also likely to be the causal variant for the blood cell trait. To test my hypothesis, I analyzed the following data. I downloaded GWAS summary statistics for 28 blood cell traits derived from more than 450,000 white British individuals in the UK Biobank. Studied blood cell traits include red blood cell count, lymphocyte count, and etc. And as I mentioned, I analyzed PU1 ChIP-seq data from 49 European individuals. The data were generated in a cell type called lymphoblastoid cell lines, or LCLs. And they are cells derived from B cells, so they could be relevant to several white blood cell traits. I first evaluated whether PU1 motif altering variants affected PU1 binding in the data. Here, I show the comparison between the predicted effects on PU1 binding on the x-axis and the estimated effect sizes of the variants on the y-axis. Each point is a variant overlapping a PU1 motif. I used a method called GKM-SVM to predict PU1 affinity of the 30 base pair sequence surrounding each variant. And the differences in the scores of the two alleles of the variants are the predicted effects on PU1 binding. As you can see, there is strong correlation, suggesting that the difference in the GKM-SVM score is quite predictive of the variance effect on PU1 binding based on ChIP-seq data. As demonstrated by the example in the middle, even for variants that change the motif sequence, if they minimally alter the predicted affinity, they generally did not affect PU1 binding significantly. Moreover, PU1 motif altering variants were more likely to be found within the center 50 base pair of the ChIP-seq peak than beyond, which implies that these variants likely disrupt directly bound sites. And I will also comment that more than half of the PU1 binding QTLs did not have a motif altering variant of PU1, suggesting possible alternate mechanisms. So I proceeded to perform the colocalization analysis. First, I collected 1,623 pairs of PU1 BQTLs and blood cell traits associated loci that showed significance to both. I applied JLIM and Coloc, which are two of the colocalization methods that make different statistical assumptions. And ultimately, there were 190 pairs that were colocalized based on both methods. they consisted of 69 different loci. And they are the loci where PU1 binding variation potentially explains the changes in the blood cell traits. Surprisingly, there were many loci that showed different results in the two methods, and this further motivates using two complementary methods to identify high-confidence colocalization. Now, I searched for PU1 motif-altering variants in those colocalized loci. Most were single nucleotide polymorphisms, and 7 were short insertions or deletions. In only 13 of the loci, shown in gray, I could not identify a likely PU1 motif-altering variant. The single nucleotide variants altered various positions along the PU1 motif. PU1 is known to also bind together with IRF vectors, which bind the GAAA motif towards the right. More variants alter the positions that are more important, characterized by bigger letters in the sequence logo, possibly because they have larger effects. Lastly, I compared the PU1 BQTL effects with allelic imbalance effects. If an individual is heterozygous, meaning that the two copies of the chromosomes have different alleles, PU1 could bind more on one of the copies. And what this result shows is that PU1 motif-altering variants that increased PU1 binding across individuals also increased PU1 binding within a heterozygous individual. Now, without this strategy, it would be quite challenging to pinpoint a likely causal variant at an associated locus. Typically, statistical fine mapping is used to narrow down candidate causal variants for GWAS data. However, we expect to find many correlated variants, as was the case with loci that colocalized with PU1 binding QTLs. Moreover, a commonly used complementary approach is to merely look for genetic variants that alter some transcription factor motif. However, there are so many different transcription factors, over 1,600 in fact, that several variants can alter numerous TF motifs. Therefore, this approach also does not usually lead to a single candidate causal variant. On the other hand, with our approach with using TF binding QTL colocalization, we focus on one specific transcription factor, and we can potentially prioritize a likely causal variant as well as the transcription factor binding that is affected. To better understand the regulatory effects of the colocalized PU1 BQTLs, I evaluated the associations to various regulatory phenotypes. I analyzed ataxic and histone ChIP-seq data from lymphoblastoid cell lines to generate chromatin accessibility QTLs and histone QTLs. Around half of the colocalized PU1 BQTLs with motif-altering variants were associated with chromatin accessibility and H3K27 acetyl and H3K4 monomethyl marks. And those QTL effects were strongly correlated with PU1 BQTL effects, suggesting that the changes in PU1 binding may affect gene regulatory activity in the locus. For these PU1 BQTLs, the PU1 motif-altering variants have a strong case of being the causal variants for the colocalized blood cell traits. As expected, some of the PU1 BQTLs were significant expression QTLs, but I will stress that there were only 10 of them that showed significance. Whether the others do not affect gene expression, or if the effects are merely too weak to detect, needs to be investigated. To finish up, I will share 2 of the loci as examples. This locus is associated with lymphocyte count, and there are more than 40 variants with similar p-values, all shown in reddish color. With the GWAS data alone, we can't pinpoint a likely causal variant. However, the signal colocalizes with a PU1 BQTL that has a PU1 motif-altering variant near the center of the PU1 ChIP-seq peak. I'll call this variant RS38. Now, RS38 increased the PU1 motif score, consistent with its association to increased PU1 binding. And the variant is associated with lower lymphocyte count. Below, I show that the PU1 binding site is at the promoter of the ZC2HC18 gene. And it also shows high level of chromatin accessibility and 3 active histone marks, H3K4 monomethyl, H3K4 trimethyl, and H3K27 acetylation marks. I additionally evaluated the effects of RS38 allele dosage on those regulatory phenotypes in LCLs. It turns out that this variant is significantly associated with all of those regulatory phenotypes. Similar to the variant's effect on PU1 binding, all showed increased activity for the G allele dosage, including ZC2HC1A expression. To investigate whether RS38 is really likely to be the causal variant, I dug through available reporter assay data. Massively parallel reporter assay, or MPRA, concurrently tests the regulatory activity of sequences that are typically hundreds of base pairs long. And we can use this assay data to test whether the sequences with one allele or the other show difference in reporter activity. Now, this plot shows the allelic effects of the sets of variants that are correlated with RS38 in this locus. And when the allelic effects are statistically evaluated, only RS38, shown in red, showed significant increase in reporter activity, which was replicated in another study. This result shows that RS38 is likely to be the causal variant among the set of over 40 candidate variants in this locus. Lastly, I also sought evidence to make sure that PU1 binding is having a role for ZC2HC1A promoters activity. A study investigated how knocking out SPI1, the gene that encodes PU1, leads to changes in chromatin accessibility genome-wide. And when I analyzed this data, I observed that there was a significant drop in chromatin accessibility of the ZC2HC1A promoter in samples without the PU1 gene. With all these supporting evidence, I'd like to make the case that RS38 and its effect on PU1 binding likely cause a reduction in lymphocyte count. One lingering mystery, though, is that the causal gene in this locus is likely to be IL-7, not ZC2HC1A. This gene, IL-7, contributes to B and T lymphocyte development, and a multi-ancestry GWAS study found a missense variant in IL-7 associated with lymphocyte count as well. And RS38 is weakly associated with lower IL-7 expression levels in LCLs. So my working mechanistic hypothesis is that RS38 directly upregulates ZC2HC1A expression due to increased PU1 binding and indirectly downregulates IL-7, possibly by taking away transcriptional machinery. However, this is only a speculation at this point. For the second interesting locus, we can see that a PU1 BQTL more than 250 kilobases upstream of the ZNF608 promoter colocalizes with a lymphocyte count signal. The PU1 BQTL has a motif-altering variant that increases affinity, and I will call this variant RS12. This PU1 binding site also shows chromatin accessibility and active histomarks. Similar to our previous example, this variant is significantly associated with increased regulatory activity across the regulatory phenotypes, suggesting its effect on gene regulatory activity. So the natural question then is whether RS12 is an EQTL, expression QTL for ZNF608. Not much is known about this gene, but there was a study that observed that follicular lymphoma patients showed recurrent mutations in the gene for about 10% of the patients in the study. And follicular lymphoma is characterized by uncontrolled division of a subtype of B lymphocytes, making it somewhat relevant to lymphocyte count. When I refer to the EQTL statistics in LCLs, the lead variant in yellow was actually elsewhere. And it was unexpected because we observed regulatory effects at the level of chromatin accessibility and active histomark levels. So I scratched my head for a bit. Then I had the idea to look for a potential secondary signal for the EQTL. Interestingly, when I conditioned on the lead variant, our variant RS12 became more significant. And this suggests that in LCLs, there are at least two ZNF608 EQTL signals. And this is further supported by applying statistical fine mapping using SUSY to the EQTL data. But what is still confusing is why only one EQTL signal colocalizes with lymphocyte count. Now we must remember that LCLs are not primary cells. They are cell lines derived from B cells, by infecting them with an Epstein-Barr virus. And this might cause LCLs to have different gene regulatory landscape from its primary B cells. Unfortunately, there was EQTL data in naive B cells to ask this question. It turns out that in naive B cells, we only see the EQTL signal corresponding to RS12. The yellow variant now has no association whatsoever. And this reflects cell type specificity of EQTLs and the importance of investigating the right cell type depending on the GWAS loci. Furthermore, throughout lymphocyte development, ZNF608 is highly expressed in common lymphoid progenitor and B cells, suggesting that this gene has a role in B cell differentiation. Therefore, I hypothesize that the lymphocyte count association in this locus is likely driven by a PU1 motif-altering variant that increases ZNF608 expression during B cell differentiation. Now, here are some takeaways from this project. PU1 motif-altering variants affect PU1 binding predictably. However, other transcription factors may behave differently. So whether this approach generalizes to others needs to be examined. And PU1 BQTLs and LCLs indeed colocalize with blood cell trait signals. But LCLs are of lymphoid lineage, making it more relevant to lymphocyte-related counts. But PU1 is upregulated also in myeloid cells. So data from other myeloid cell types could lead to further discoveries. And blood cell trait-associated PU1 motif-altering variants affected many regulatory phenotypes. However, only 10 showed significant expression QTLs. The remaining cases could just be weak signals that are hard to detect, or the variants that are affecting gene expression in a different cellular context. Finally, I showed that transcription factor binding QTL data can be used to pinpoint likely causal variants in GWAS data. The challenge, though, is selecting the right TF and cell type for some trait or disease because generating data always costs money and time. In my opinion, starting with a master regulator like PU1 seems like a good strategy. With that, I thank Martha for guiding me throughout my PhD and encouraging me to explore statistical genetics, even though the rest of the lab was wet lab. And I also thank the past and present lab members who have given me insightful feedback. Thank you very much for listening.
Great. Thank you so much for that excellent talk. Now we have some time for some questions from the audience and the panelists. And as a reminder, there's no questions now, but feel free to just post those questions to the Q&A. And we'll go ahead, I'll read them out to the speaker. While we give some time for some questions to come in, I just have a couple here for you to get you started. So I found the IL-7 hypothesis very, very interesting, and I was just kind of curious if you have any plans or ideas of experiments that you could do to explore this hypothesis of how it's downregulated.
So yeah, I also found this locus very interesting because of its weird direction of effect. Currently, we don't have clear plans of testing this hypothesis. And I'd be like in the future, I'd be interested in looking for other loci where something like this is happening. Like we typically think genes that are close by are co-regulated. So if one is upregulated, the nearby genes are also upregulated by the genetic variants. But this is, I guess, a counterexample to that. So whether this is truly the causal effects of the same variant that affects PU1 binding or not, I think would be very interesting to follow up on.
For sure, for sure. And then I think you did a little bit of this, but I was kind of curious if PU1 is expressed in most blood cells, and if not, maybe you could kind of use that to try to corroborate some of the associations you found for cell type specific effects.
So yeah, for one, PU1 is not always expressed throughout differentiation. So like for even lymphocyte differentiation, it is upregulated for B cells, but downregulated for T cells. And for red blood cell differentiation, it is downregulated. And for myeloid cells, they're, I think, mostly upregulated throughout the process. And that the detecting the cell type specificity would involve both just the identity of PU1, but also the cell type in which this ChIP-seq data was performed on. And I'm sure that some of the loci, some of the regulatory effects of PU1 binding that we find in the lymphoblastoid cell lines are shared with some of the progenitor cells, may perhaps related to more lymphoid differentiation, but may also share some regulatory landscape with myeloid cells. In fact, some of the colocalized loci corresponded to like monocyte count or eosinophil count that was of myeloid lineage. So in those examples, the regulatory effects might be shared, but I would expect. So I guess to add to that, the number of colocalized loci in this study only explains a small subset of all blood cell trait associated loci. And likely because PU1 is not the only transcription factor in blood cells. So there could be many other factors that are affected by the other genetic variants. But also, yeah, LCLs are just one specific cell type that limits to what we can find by colocalization.
Yeah, great. So we have a question now from the audience. So first off, they say, thank you for the great talk. And then they ask, can you elaborate a little bit on how EQTLs are likely to be BQTLs in general?
That's a good, interesting question. So a typical hypothesis for genetic variants affecting gene expression is through, if they're in the non-coding region, is through affecting transcription factor binding. So if we have data of the correct transcription factor for a specific locus, I would expect that most EQTLs would also be BQTLs for some transcription factors. If we, although I only showed a plot of PU1 binding QTLs that colocalized with blood cell traits, only had 10 of them that showed expression QTL effects. I don't know the actual numbers, but when I compared it with all of PU1 binding QTLs, more were expression QTLs. So there's definitely, it makes sense that a variant that affects transcription factor binding would affect gene expression somehow. But whether they are all detectable at a current sample size is also an issue. Like for instance, we expect most non-coding variants to affect gene expression in the loci that are associated with complex traits, but we only find a limited number of such colocalizations. So there's both issue of statistical power with the current sample sizes, and also just the potential limitations of the colocalization methods in its sensitivity.
Great, yeah, and just one more question here. Adrini was curious if you remember if you happen to see any colocalization between sickle cell associated variants and PU1 binding. You might not remember off the top of your head.
Oh, yeah, I did not investigate that question, so I don't know. But I imagine, so in this work, there weren't that many red blood cell related traits that showed colocalization. Probably because for red blood cells, PU1 is downregulated throughout that process. So maybe looking at other transcription factors might lead to interesting findings related to sickle cell disease. But in this project, I would not expect to find that many examples because PU1 is downregulated in red blood cells.
Okay, great. Yeah, thank you so much. And just wrapping up now, I want to say thank you again to both of the speakers for their excellent talks. And thank everyone for attending this month's VES. And be sure to tune in on December 5th when we'll have another variant effects seminar. So thank you, and we'll see you next time. Bye.